Awesome, thank you. All right, so yes, ATP, we said, is the currency inside of our cells. It's like dollars, if you will, that can be spent to do the activities, the processes that our cells need to do. If we look at the molecule ATP, it's an adenosine molecule, and then it's got 3 phosphates attached to it. So it's adenosine triphosphate, right? 1, 2, let's get my pointer. All right, so it's got 1, 2, 3 phosphates attached to this adenosine in order to make ATP. And so energy from exergonic reactions is used to make ATP so that we can then have it on tap, have it on hand as currency to be spent inside the cell, okay? In other words, our body makes ATP just so that it can have it ready to go when we need it. Think about having a backup charger for your phone. The reason that you charge it is so that you can have it ready when your phone dies and you're out, on the road or whatnot. So very similar scenario. We make ATP in order to have it ready so that we can spend it or use it to do work inside the cell. Let's talk about how we actually make that ATP. So in order to do that, we need to have a phosphate plus the energy that we're using, right? So it's going to take the energy that's released or liberated from a reaction, and then we can add that phosphate onto ADP and make ATP. And as a release, as a byproduct of this reaction, we're going to release some water as well, okay? Now the change of energy is about 7 kilocalories per mole. In other words, this is an endergonic reaction. It's going to take some energy to do this, right? So if you think back to this initial equation up here, we had to invest some energy here. That is why we can describe this as being endergonic, right? Same scenario, when you charge your backup battery at home, you're putting energy into the charger so that you can have it later on when you need it. We can also consider this to be a condensation reaction. Anytime we make water as a byproduct, it's going to be a condensation type of reaction. And then there are 2 ways that we can do this inside our body. There are 2 general ways of adding that phosphate onto ADP in order to make ATP to be used later on. Now, the first way is called substrate level phosphorylation. And so what that does is it takes the phosphate from another molecule in the cell, kind of steals that phosphate or borrows it, if you will, from another molecule. In this instance, it's molecule X. We're going to take the phosphate from molecule X. We're going to also take ADP. We're going to, again, invest some energy here, and then we're going to adjoin that phosphate onto ADP to make ATP, and then we're going to be left back at the end with the other molecule who now has lost its phosphate. Okay, so this is called substrate level phosphorylation, and it's synonymous with taking or borrowing a phosphate from another viable molecule in the cell. The second way that we make ATP, and this is the more common way, this is the more reliable way, this is the more efficient way to make ATP, and that is going to be the electron transport chain, which is where we phosphorylate ADP and make ATP. And this is a more sustainable, viable way of making ATP. And we're going to talk about how we use the mitochondria to do this. We use the transport of electrons across the mitochondria in order to generate the energy that's required here to make ATP this way, okay? Now, as the name implies here, oxidative, this is going to require oxygen, okay? Substrate level phosphorylation does not require oxygen. It's kind of a cheap, fast way to make ATP. It's going to have a price to pay that we'll talk about later on, but it's an easy, quick way to grab another phosphate off of an existing molecule and make a smaller amount of ATP this way. Oxidative phosphorylation is going to require oxygen, it's going to require the mitochondria and the electron transport chain, but it's going to make a lot more ATP, and again, it's going to be the more reliable, sustainable way to make ATP. Okay, let's talk about how we break that ATP down on the other end. So as I mentioned earlier, ATP is made only to be broken. It's stored only to be utilized, right? So it's a temporary energy storage for our cells, for our body. And the way that we break it down is going to be a hydrolysis reaction, so the very opposite to that condensation reaction. So on the left of our equation, in this case, we have ATP. We're going to have to invest water here, right? It's a hydrolysis reaction. We're going to have to lyse or cut or open this water molecule to liberate that water. And then we're going to make ADP and then phosphate, and then we're going to liberate that energy on this side of the equation in this instance. Okay, this is the energy that we're interested in. This is the energy that we want for our cells to utilize in order to do various cellular processes or cellular work. Okay. All right. Let's think about the central reaction equation for the metabolism of energy. All right. So what that is glucose, right? C6H12O6, which is glucose, a molecule of glucose. Oxygen, that's going to be required as well. And then that will give us carbon dioxide, water, and energy. Okay, carbon dioxide, water, and energy. This is how our cells can take the food that we eat, i.e. glucose, invest oxygen. Remember, again, we've talked about how cellular respiration requires oxygen, right? It's an oxidation type of reaction. And then we're going to liberate carbon dioxide as a byproduct or waste product. That's the carbon dioxide that we breathe out or exhale in our lungs. We're also going to generate some water here, and then we're going to generate some energy. Again, the energy is the important part here. This is the energy that we're relying on for our cells to do work, right? And the change in energy of this reaction is about 686 kilocalories per mole. Okay, this is the amount of energy that was liberated from one molecule of glucose. All right. And I want you to kind of think back to this reaction and think about this reaction being driven in the reverse direction. So think about energy being invested, water being invested, CO2 being invested, and then we make glucose and oxygen on the other end. What reaction is this if it's happening in the reverse direction? Who can identify that? So energy, water, and CO2 is being used, and oxygen and glucose are being released on this end. What's this an example of? Think back to your biology courses, your foundation of biology courses. Jog your memory back to those topics and see if this reminds you of anything else that you talked about. Okay, so this is an example of photosynthesis, right? So plants can do this reaction in the opposite direction. And this is a really good way to highlight that symbiotic relationship between our body, between humans and plants, right? So plants can take the sun's energy, water, and carbon dioxide that we breathe out, and they can convert that into food and oxygen, right, that we also need. So this is how we can release the things that plants need, and plants can release the things that we need. And this is how there's that symbiotic relationship happening that's expressed in this reaction. We can do it in the forward, and plants can do it in the reverse direction, right? So really interesting to make a note of this reaction. It's happening in our cells in the forward direction, and it's happening in plants in the reverse direction, okay? All right, let's look at this reaction in a different way, okay? So here's glucose oxidation. Here's what we just described on the previous slide. So we take one molecule of glucose, we have oxygen, right, that we're breathing in. we're going to liberate energy by lysing that molecule, and then we're going to end up having carbon dioxide and water released as a byproduct. And then in looking at the energy that we release, we're going to be able to use that energy to drive that phosphate onto ADP in order to make ATP. Okay. Now, I want to point out that in doing so, we also liberate some heat. Okay, because we are not completely efficient beings, because we do have thermogenesis happening in our body, some of the energy that we liberate, all of that doesn't get captured to make ATP. A lot of that just gets released as heat, okay? So this is why our bodies can generate heat, which we actually need as a part of homeostasis. But I really just want to highlight that this is not a perfectly efficient process. Our bodies are not perfectly efficient beings. Most of the energy that we capture when we break down glucose is used to make ATP, but some of that energy is escaped into our atmosphere, and that is liberated from our body in the form of heat. Okay, so after we do make this ATP with the energy that is liberated, or some of the energy that's liberated, we can store that ATP in various ways in our body, in our muscles, in our cells. And so we'll have it on tap. We'll have it just kind of hanging out here waiting to be liberated again in the form of cellular work. So things like movement of molecules throughout the cell or in between one cell and another. Things like making things, making proteins, making hormones, right? Making our phospholipid membranes, making the things that are required inside the cell as well as outside the cell. Also transporting some of these molecules and components outside the cell across the membrane so that they can be moved around in the blood or in the body in general. Okay, so these are the types of activities that we mean when we say that energy is required to do cellular work in our body, right? The cellular chores, the things that the cell has to do in order to keep us alive. Right, and I want to go back to that example that I gave of your backup battery pack. This is the backup battery pack, right? This is you charging it so that you can have it on hand, but it's only charged so that you can use it to do the things that you need it to do when it's time to do so. All right. So here is this reaction once again. So we talked about the reaction. Let's add some more values or some more numerical components in here and talk about how this is not an efficient process, right? We illustrated that on the previous slide, the fact that this is not a perfectly efficient process because we lose some of that energy as heat, but let's actually validate that by talking about the values. So here we have 38 ATP that is made when we liberate a single molecule of glucose. So we've got a single molecule of glucose. We had to invest 6 molecules of oxygen, and we had to have on standby 38 molecules of ADP. And we're also going to require 38 phosphates in order to phosphorylate 38 ADP. On the other end of our equation, we make 38 molecules of ATP, and we free up 6 molecules of carbon dioxide and 6 molecules of water as a byproduct to this process. All right. Now, if we think about the ATP that we invest, the ATP that we yield from this reaction, that gives us about 266 kilocalories. Okay. If we multiply the amount of ATP molecules, that are 38 moles, by the amount of energy that is possessed in each of those moles, we're going to end up having 266 kilocalories per mole. Now, if we think about the amount of energy that's released during glucose oxidation, when scientists look at this process, what they see is that, in fact, 686 kilocalories should be released, right? We should have, in a perfect process, we should have 686 kilocalories being released for a single molecule of glucose. all right? So essentially, there's a discrepancy between the amount of energy that we do actually yield and the amount of energy that we should yield in an ideal or perfectly efficient scenario. So the difference between the energy that we make and the energy that we should make is 420 kilocalories. All right. And so what this tells us is that glucose oxidation is only 38% efficient. We only yield 266 kilocalories out of 686 kilocalories that we should optimally yield. And so only 38.8% of that is yielded. And so as a result, we lose 61.2% of that energy as body heat, okay? So all of this really just to say or to illustrate what we said earlier is that glucose oxidation is not a perfectly efficient process. Most of the heat, 61.2% of the energy that we make is liberated as heat, and only 38.8% of that energy is actually used to make ATP for energy for our cells. Okay. All right. I'm going to take a pause here. We're almost out of time, but I want to take a pause and see what questions we have on what we've said so far. Okay. Don't see any questions popping up. All right, let's go ahead and keep going here for a few more minutes. So let's talk about the stages of glucose oxidation. That's what we're going to get into next. And this is essentially the steps that are taking place in order for this reaction to happen. Okay, so this is the reaction that we're focusing on. This is kind of the primer for what we're going to say next, right? What we're going to go on to discuss next is that we have one molecule of glucose, we have oxygen, we have ADP, we have phosphate, and in order to drive this glucose oxidation reaction, it's going to take a series of steps that we're going to talk about in detail next, okay? Those steps are glycolysis, the Krebs cycle, and the electron transport chain, okay? What we want to further say here is that two of these stages, glycolysis and the Krebs cycle, are going to make ATP via substrate level phosphorylation. Who can remember what substrate level phosphorylation was? What was the analogy that we used earlier? What was the analogy or the example that we gave earlier? Anyone remember what we talked about? So if we think about this molecule being a molecule that's phosphorylated in the cell, ADP on this end of the equation, and then we end up having the molecule now lost or losing its phosphate, and then ATP made on this side of the equation. So the analogy that we gave earlier was if you were to borrow a phosphate or grab a phosphate from another molecule. So in glycolysis and in the Krebs cycle, ATP is made, right? Phosphorylation of ADP happens via substrate level phosphorylation, where phosphate is taken from another molecule, added on to ADP in order to make ATP. Now, the final stage of glucose oxidation, which is the electron transport chain, is where we use oxidative phosphorylation. This is where we use the transport of electrons in order to add that phosphate onto ADP and make ATP. So this is what we describe as being the way that the majority of ATP is made in our cells. It's also the more reliable mechanism for making ATP, but it's going to take a longer amount of time and oxygen is going to be required as a substrate in order to drive this reaction.